Next, we're going to hear from David Reich from the Department of Genetics at Harvard University. Oopla.
So I'm going to be talking about work that was led by Svante Pääbo in the Max Planck Institute in Leipzig and by Ed Green, who's a postdoc now at UCSC, and a project I've been involved in for the last few years. I'm going to begin talking about a little bit of background about the Neanderthal, who Neanderthals are, based on the paleontology and the archaeology. I'm going to talk a little bit about the sequencing of the Neanderthal genome that was done by the Pääbo group. And then I'm going to talk about 3 lines of evidence for gene flow from Neanderthals into modern humans, which we think might be explaining the patterns that we see in the data. And then I'm going to do a little bit of a conclusion, maybe talking about possible implications for phenotype and medical genetics today. So the background I'm going to be talking about is not work that is background about who Neanderthals are, as defined almost entirely by archaeology and the fossil record, and the draft sequence of the Neanderthal genome that was generated by the Pääbo group, and was not work that I did, but was work that's very important to understand in order to understand this data. So the important thing to realize about Neanderthals is that they are our contemporaries until 30,000 years ago. Modern humans and Neanderthals both first appear in the fossil record in a sort of late form, in a fully developed form, by about 200,000 years ago, although there's a debate about the exact date. And modern humans evolved in Africa. The oldest fossils of anatomically modern humans we have are about 200,000 years old from Ethiopia. But they also, the Neanderthals are from Western Eurasia. What's very interesting is that after 130,000 years, modern humans appear outside of Africa for the first time. They're initially in the Levant in the Near East, and then there's an explosive expansion out of Africa by about 45 or 50,000 years ago, where modern humans leave the Near East and they go to Europe and they go to Eastern Asia and they go to Southern Asia and they go to Australia. And in this period between 130,000 years and 30,000 years ago, there was places of overlap. They're best documented in Europe between 30 and 45,000 years ago. Here are pictures of archaeological sites. Round is modern humans, square is Neanderthal, and they also overlapped in the Levant between about 40 and 135,000 years ago. In particular, they overlapped near the Mount Carmel region of modern Israel. And it's very interesting because whereas in Europe they were contemporaries of each other, they were right next to each other, so they must have interacted between 30 and 45,000 years ago. It's never been documented that they were there at the same time in the Levant. The first modern humans get there about 100 or 100,000 or a little older years ago, and then there's a gap, and then there are Neanderthals at 80,000 years, and then there's a gap, and then there's modern humans at 60,000 years, and there's archaeologists and paleontologists have debated about whether they ever interacted or whether the movement of populations was just due to glacial and climatic changes. So because of these regions, this evidence of interaction, the question is there interbreeding that occurred between Neanderthals and modern humans? And there would have been many possible places for this to occur. It could have occurred in the Levant between 30 and 130,000 years ago. It could have occurred in Europe, where there's really well-documented evidence, or the archaeological evidence isn't as good, but maybe further in Eastern Asia. So the Neanderthal Genome Project, which, as I said, was led by Svante Pääbo and many colleagues in the Max Planck Institute in Leipzig, did an enormous amount of work just to find the DNA samples that were studied in order to sequence the Neanderthal genome. This involved screening making 89 extracts from bones from many sites around Western Eurasia, especially Europe. And most of these extracts don't even produce any DNA. The bones are very old, typically 30 or 40 or 50 or 60,000 years. No DNA is extracted. If DNA is extracted, it's often all microbial. It's fungi and bacteria, and there's no evidence of primate or ape DNA that's convincing. And even if there is evidence of primate and ape DNA, there's a serious worry that it's contamination of modern human DNA. And this is a very serious problem. It's affected past studies. And it's particularly serious because the reads you get are short and degraded, only typically 40 base pairs long. They're so short that you can't tell whether they're modern human or Neanderthal. The typical number of differences between modern human and Neanderthal is about 1 in 600. And so if 40 base pair read is most of the time identical to a modern human, you can't tell whether it's Neanderthal or modern human. So The DNA sequence for this study is mostly from one place, from 3 individuals from Vidya Cave in Croatia, but there's a smaller amount of sequencing. The largest amount of it is from Mezmaiska, from a child from the Caucasus in Russia, and also from Spain and from Germany, from the Neander Valley, where the type specimen of Neanderthals was found. So the way the DNA sequencing and extraction was done was the scientists drilled beneath the surface of the bone with a sterile dentistry drill. The idea is to avoid contamination at the surface, which might be more likely to affect the surface. Bone powder is extracted. It's sequenced on a 454 or an Illumina sequencer. It's mapped to the ape genomes, to the human or the chimpanzee, and the sites that map to human and chimpanzee are used. In this data set, only about 3 to 10% of the data matched to apes, and that was after an enrichment procedure. So most of the material is microbial, and there's a great deal of sequencing that's done just to obtain the 1.3-fold coverage that was eventually obtained. The genome sequence is really very, very drafty. It's maybe we can't even call it a draft. About half the bases in the genome aren't even covered once, and what is covered is full of errors. An important issue with this type of DNA, as I mentioned, is contamination by modern human DNA. This has affected previous studies, as I mentioned, and is very important to consider in order to understand whether you can make population genetic inferences about the evolution of modern humans and Neanderthals and possible interactions. So since in the last few years, the PAPO lab has implemented a number of rigorous procedures in the lab in an attempt to minimize, but you can never completely eliminate contamination. And these involve adding a barcode to the fragments of DNA extracted from the bone when they're right at the time of extraction. So there's drilling into the bone, the DNA is extracted, you get these 40 base pair fragments typically, and you add a unique barcode to the Neanderthal sequence, and then you add the 454 or the Illumina adapters, and ever after you require to see that barcode if you're going to use the data. And this reduces or eliminates contamination after the library production. But before the library production, for example, contamination by an experimentalist or an archaeologist is not eliminated. We also looked at mitochondrial, Y chromosome, and autosomal methods to estimate the contamination rate. Basically, there are sites that are distinguish all the Neanderthals and all the modern humans that have been seen so far in the mitochondrial DNA, and the mitochondrial DNA occurs in high copy number in the genome, and so you can see that there are many more matches to the Neanderthal than to the human, but not all Neanderthal, and so that results in a contamination rate estimate of about 0.25%. If the male individual you're sequencing is a male, you expect this number of sequences from each of these 3 bones, but you see many fewer, so these are 3 female bones. And there is a little bit of male sequence that you see, and so the estimate of contamination rate here from the male contamination into these female bones is about 0.5%, and there's an autosomal estimate, which is again less than 1%. So looking at the data, it looks like there's less contamination, less than 1% contamination, but not 0. And so we need to be cognizant of it, but we're excited that it's small. And so ever after, when we try to do population genetics to study the relationships of the Neanderthals and the modern humans, we try to be cognizant still of the possibility of contamination, because we're trying to look for evidence that Neanderthal might be more closely related genetically to some human populations than others. So contamination could produce exactly that kind of effect. So you need to be very worried about it. So the first line of evidence for gene flow that we found was very simple, and it's that non-Africans are genetically closer to Neanderthals than Africans are. And the way we do this is we take a very simple experiment. We take humans from 2 different populations who we've sequenced, and we ask, does the Neanderthal match one more than the other? So you compare, say, a French individual or a Yoruba individual from Africa. You call SNPs between them based on single haploid reads. You identify differences between them. And you restrict to sites where Neanderthal has the derived allele. So if it's a TG SNP, you ask, does the Neanderthal have a T or G? And the reason we're restricting to cases where Neanderthal has the derived allele is because that means that we've seen both alleles twice. The Neanderthal has the derived allele, so the chimp has the ancestral allele, one of the modern humans has the derived, one is the ancestral. The likelihood of seeing the derived allele twice due to sequencing error is much less than only once due to sequencing error, but not totally negligible, but much less. So another thing that we try to do is we've mapped everything to the chimpanzee genome, not to the human reference sequence. We thought that was important because the human reference sequence is of a particular ancestry, and maybe that would bias our analyses, but we think all modern humans are equally closely related to chimpanzees. What you see is 92,000 matches to the French individual and and 84,000 matches to the Yoruba. That's a hugely statistically significant result. It's 10 standard deviations. When we did this analysis for many pairs of individuals from long-read sequencing by Kidd et al., you see that when you compare two West African individuals, there's no significant differences. When you compare two non-African individuals, there's no significant differences. But when you compare an African and a non-African, in every example compared, there's about a 4 to 5% skew in extra matching to non-Africans. This is seen also when we sequenced with short-read sequencing, Illumina, a larger, more diverse set of populations, not only the HapMap-like populations, French, Han, and Yoruba, but also San Bushman and Papua New Guinea. And again, you see all the comparisons of non-Africans to Africans have this 4 to 5% skew, but not seen in comparisons within Africa or outside of Africa. It's consistent by base substitution class. It's consistent based on the 454 and Illumina sequencing transitions and transversions with each bone that we looked at. And so it seems to be a robust result. It's consistent across the X chromosome and the autosomes.